Hey y'all, the last major mechanism that we're going to be talking about is genetic drift. Genetic drift is probably one of the toughest mechanisms to sort of wrap your head around, mainly because it has a little bit more to do with probability than some of the more straightforward and obvious forces that we've been looking at so far. So genetic drift is random as a mechanism, and its definition is that it's a change in variability due to chance. So just like natural selection, it changes variability by decreasing it. So that means that rather than having, let's say, 50-50 of different types, we're moving further away. So we have 70-30, or there's more of some than another. Unlike natural selection, however, there's no reason for it. So we see these fluctuations away from perfect variability just by chance, not because anything has a better fitness than anything else. And so again, this means that it's random because since we can't predict anything in the population in terms of how drift will affect it, it isn't the same as natural selection where we could actually predict which things would increase or decrease. So one of the ways that drift can occur is from outside forces. So natural disasters are often an example of this. So if you think about, as our walk through time in history, there were some major natural disasters like volcanoes and meteors, and they would randomly affect the biodiversity in the area. So a smaller version of this is the picture on the right up here of what I call the Bigfoot in the sky here. So let's say at the beginning that there are equal numbers of green and brown bugs. And they reproduce the same, they have the same fitness, there's really not much different about them. They're equally good at avoiding predators, but just by chance, someone comes along and steps on mostly the green bugs. So this wasn't any reason for it, he wasn't looking, it wasn't on purpose, just stepped on the green bugs. And so that means that there are more brown bugs left. And so then that means in the next generation, there's a high probability that there will be more brown bugs instead of an equal number of both. To understand the other way that we talk about drift occurring, you have to know a little bit more about genetics. So there's a few definitions here to go through. The first one is alleles. So the word alleles is another word for different versions of a gene. So for example, if I label a gene A, it could have version A1 or A2. It could have A3 or A4, but we're gonna stick with just two versions for right now. Now you, if you look down a little bit further, are a diploid organism because humans reproduce sexually. So that means that from a biological standpoint, our genes come from a mom and a dad, so a maternal source and a paternal source. That means that we can be, for any gene that we have, either heterozygous or homozygous. So we have two copies of every gene. If they're the same allele for those genes, we would consider that homozygous. And if they are two different alleles for that gene, we would consider it heterozygous. Our sex cells for humans are haploid. So things like sperm and egg, because they have to come together to make a whole, they have to start out half as big. And so those are half sized, or we would consider them having one source for each of their genes. This won't come into play too much right now, but a good definition to know for later. Okay, so to use this example a little bit further, we're making this diploid offspring called Circly. So Circly is heterozygous A1, A2, and that is because Circly received an A1 from mom and an A2 from dad. So as a diploid heterozygous individual, when Circly reproduces, there's a 50-50 chance of Circly passing on A1 or A2. So he can pass on one or the other, but not both. And so this 50-50 probability is very important if we look at other heterozygous individuals in the population. So right now we're looking at one individual, kind of one family lineage, but let's say that there are a lot of heterozygous circle organisms in a population. So in this picture we have 10 circles, okay, so just like Circly, that are A1, A2. Now, just by probability, because each one has a 50-50 chance of passing on A1 or A2, we would expect about 5 of the 10 to pass on A1 and 5 of the 10 to pass on A2 into the next generation. So if the probability is equal for A1 and A2, we'd expect it to be an equal outcome. What genetic drift says is that just by chance, and genetic sampling, what we consider error, just variability, 7 of these heterozygous circles could pass on an A1 instead of an A2. And so our percentage instead of 50-50 is 70-30. And if you consider that, if all of these blue ones pass on an A1, then their offspring may also have a higher chance of passing on an A1. So you may see an increase in A1 over generations because of just chance variability. So to look at how this applies through generations, we have to look at more than just a handful. So a good way to do this that we sometimes will actually try in person if we're in person together learning is to fill a bag with beads of different colors. So in this bag, there are 100 beads, and they represent the A1 and A2 alleles. So orange is A1 and green is A2. So let's say we started 50-50 like we did in our last example, and we draw 10 out just to represent a sample to predict which alleles will be passed on by these 100 individuals. Okay, so if we pull 6 and 4, okay, so 6 A1s and 4 A2s, 6 orange, 4 green, then that tells us that our variability has changed from 50-50 to 60-40. So now we have 60 orange and 40 green. And if we again pull 10 to represent our population, we're going to pull 8 orange and 2 green. Okay, so we can see that it went up in terms of the number of orange in the population. And then we repeat it, but this time we pull 10 orange. So this would say just by chance, every one of those heterozygous individuals passed on orange instead of green. And so this means that 100% of the offspring have this orange A1 allele. And so that means that green allele is gone. It's gone permanently. So there's no probability for it to come back. And so when we say that genetic drift can cause you to lose an allele, this is the kind of example we're talking about. But this is random, so it doesn't always follow that trend. So if that was one of my groups that actually did this experiment at their table and working with beads, this could be another. So our first generation hit 60-40 again, but when we drew our 10, we got 70-30. So we did see a slight increase in orange over green, but then we go to 4 and 6. So instead of continuing to go up, our orange numbers, our A1s actually went down. Because again, this is random. It has nothing to do with selection or reproductive success, it's just by chance. And so you can often see this fluctuation up and down in different populations. One of the things to note though is that this fluctuation, so we can see here, is fluctuating up and down. In this example, we lost an allele completely because we were drawing only 10 reproductive individuals from the population. So that's really relatively a small sample. So when we look at sample size, if you look over here, this is an example, a sample size of 4. If we assume that we start with 50-50 of our A1, A2 alleles, and then each one of these lines represents a population. So if we send each one of these lines is like one of those bags that we're letting, but with only 4, that we're pulling randomly from to see which ones reproduce. What you see is that very quickly we go to what we call fixation. So if gray is representing one allele, the gray lines down here representing one particular allele, A1, then a bunch of these lines have gone to fixation or have been lost completely in only 20 generations. When you see them up here at 1, that means they went to 100% A1, so they lost the A2 allele completely in 20 generations. However, if you look at a slightly bigger population, we start with the same 50-50, and these are all the generations. So we're pulling randomly out of the bag to see what happens. At the end, you can see that some of them do go to fixation. So there's a couple lines that do hit the top and the bottom, but a lot of them just continue fluctuating up and down. So the likelihood of losing one allele is much less. And then last, you can see this most clearly with this population size of 400. So again, there's a lot of fluctuation, but none of them out of this group actually reach the 0 or the 100% line. So this tells us that genetic drift is a weaker force in larger populations, but a much stronger force in small populations. This also contributes to something called the founder effect. So the founder effect isn't necessarily the same as genetic drift, but it has sort of the same starting premise. So if you look here in our population of bugs, we have a big diversity. Now, when we did our walk through time, we looked at how the continents moved away from each other and like islands were forming. So let's say that this group here was on a piece of continent that broke off and became an island, or just moved far away as its own continent. Because this is the starting population, we're limited in what traits the next generations or the descendants can have. So we only have the gray and the yellow bugs. We're missing the orange and the red bugs. And so that means that in this area, only the gray and the yellow bugs will increase. However, let's say we make another island or a continent that moves, and this founding population is different. So it's red and orangish and yellow. And so then this population will end up looking very different than this one. And the other aspect to think about is these founding populations are pretty small. So over time, they will grow to get a lot of descendants, but if they don't, they're also going to be very subject to genetic drift. So the founder populations can be very subject to drift. And it also determines basically the future of what variability will look like apart from mutations or anything like that contributing to more variability. Okay, so genetic drift, just to compare it, so we see how it fits in with these others. Remember that natural selection decreases variability, but for a particular reason. So there's a selective pressure that causes one variation to reproduce better than another. Genetic drift causes a decrease in variation, but it's just by chance. So you end up with one in a higher proportion just because of chance, either outside force or genetic just variability. Mutation is where we can get new traits forming. So this is the start of any new variability in a population, and gene flow is how traits can move from one population to another. So that is it for our mechanisms, and we will use these mechanisms for some other information as we move forward.